I'm Molly Starback, Director of the Duke Office of Postdoctoral Services. Thank you for coming out on this rainy morning. Today we're pleased to welcome one of our favorite speakers, Dr. Bill Schrader from the National Institute of Environmental Health Sciences. Bill is the Deputy Scientific Director at NIEHS, and his career has spanned academia, industry, and government. He's worked extensively with graduate students and postdocs, both in academia and now in government, and today he's going to tell us how to give a great job talk. Dr. Schrader's built in plenty of time at the end of his presentation for your questions, so please feel free to ask those. Without further ado, Dr. Schrader.
Thanks, Molly. Well, it's good to be here. First of all, before we get rolling, how many of you are postdocs? Raise your hand if you're a postdoc. How about graduate students? Okay. So this talk is kind of generally geared toward going to actually get a faculty job or a job in a company or something. But what I want to say about trying to make an impression that highlights your good points falls just as well if you're going to go and interview for a postdoc position. So this is not limited. A lot of what I'll say is specific to the people who are trying to pay you to do it, but going for a postdoc interview carries a lot of the same stories. So let's get going. By the way, I also don't mind being interrupted if it's a salient point, something you don't understand. Please just raise your hand and I'll try to call on you, take brief questions, and I'll try to give you a brief answer. Second point is if you get bored or you get a text message from your significant other and need to leave, I don't care about that either, so just get up and adios. All right, moving along in this bombastic review. I'm going to try to kind of make you street smart about some things which you, I expect, haven't really thought about. And that is what it says here. And a lot of people think that giving a job seminar is a scientific talk. Well, it is, and I'll try to tell you about that. So first of all, remember that this is a little different. Can these lights be turned off so that this is not on the screen? I'm always afraid to touch one of these things for smoke.
I think if you hit right next to it, the third button down.
Cool. Way cool. There's a man who's been in this room before. Okay, the job is to get the job offer. That's the objective, and it requires that you exchange information between you at the podium and the search committee who's invited you, and the audience who's there to listen to you, and the interviewers who you're going to meet, and also the search committee at the end after you've already spent the day with everybody. And they all have different things that they're trying to tease out of what you have to say and how. And finally, after they make all their recommendations, the hirer has to then make an informed decision, usually having met with you, heard you, etc. But they also weigh all this input that they get from everybody else. So if there's all these people on this thing, send the hirer mixed messages, it's not as well as if they all say, boy, you got to get this gal, she's fantastic. That's a big difference from, yeah, you know, not sure. Okay. So, there's a search committee formed for anything, except if you go to get a postdoc, it may just be the person who's looking for a postdoc in that person's lab and the people who are already there in the lab. But other than that, there's usually, there's almost always an actual search committee. And there are a couple of points I want to make about that search committee. They don't have a clue about your field of work, or at least most of them don't. They've been stuck on the search committee by the chair. Few of them know who your boss is. Few of them know your techniques, and none of them know your jargon. So if you get up and start throwing the acronyms and terms, by using the Berkowitz reaction or something, and nobody knows who the hell Berkowitz was, you lose. So that's a very important thing to keep in mind. You're trying to telegraph this to a mixed audience, not the people in your lab group when you give a Friday seminar what you did last week. And they have defined criteria to meet First of all, they want to know exactly what technology you're going to bring. If you notice in ads, if you've already started, how many of you have already started looking at job ads in the back of Science or somewhere on the websites and stuff like that? Okay, so you know when you read one of these things, it says, if it's a university job, you know, able to teach a course in animal cell physiology, with specialization in techniques and specifically in siRNA screening or cells, embryonic stem cells, whatever it is. And then it'll usually say something about some technique. Well, the reason is they want you to bring this capability to that company or to that department. All right? They want to know what you're going to start what you're going to do to start. Now, if it's a company, they are going to hire you into a project. They have a project to find drugs to cure hangnail, and you have some work it's an assay for hangnail in a C57 Black 6 mouse, and they want you to come in and set that assay up and run it. Okay? If you get offered a job to go and set up a C57 Black 6 assay to look at hangnail in the mouse, turn it down. Now, what head count you will inherit? Well, if it's a university job, they're going to offer you, the startup package will include a bunch of money, and generally you'll hire a technician and probably a postdoc, and then you'll try to attract some undergraduate volunteers, a graduate student or something like that. What scientific resources you will need. If you do work that involves NMR spectroscopy, they already know you need access to an NMR machine. They're not expecting you to bring that. Pick any other piece of machinery that you use during the course of your research. They know that that's what you need, or you need to make it clear to them. All right? Whose former lab space you're getting? If you're going to a company or to a university, usually somebody was already in that space. Junior faculty don't generally move into brand new space. Senior faculty move into that, and you get their old lab. In a company, you may be inheriting somebody's space, and that person may still be working there and not realize they're about to be fired in favor of hiring you. So things can happen quickly in a company setting. All right, so what's the most important thing I can tell you is that you have to stand out in the crowd. And what do I mean by that? Are you, particularly if you come out of some, chairman's, somebody here at Duke, some well-known professor or member of the National Academy or chairman of a department or somebody with 15 postdocs and 8 graduate students and half a floor. Well, are you just some little minor cog who sits there and waits for the boss or somebody to tell you what to do? Or are you the person with the little gray cells? So what exactly did you do on this project? Now, as I'm going to say in a few minutes, this means not saying we did the following experiments and we found the following result and it has been shown that. Because when I say that, you haven't a clue, I'm in the audience, you haven't a clue. Who did it? Who thunk it up? Who decided that that's what that experiment meant? Did you think up the ideas or were you just told what to do? Okay? Did you collaborate? Well, you say, well, what difference does that make? In real estate, the story is location, location, location. In companies, it's teamwork, teamwork, teamwork. And collaboration is the name of the game in corporate research. And more these days, it's also true in university research. As projects become bigger and more complicated, they need collaboration amongst a lot of people with a lot of different skill sets. So you need to demonstrate during the course of your talk that you have collaborated with other people to get work done. And I'll talk about that a little bit more later on. Did you ride everybody else's coattails? What do I mean by that? Well, sometimes you march into a new postdoc position or you take, or whatever it is, and You know, you've taken over a project that was already nearly completed, and you lucked into this knockout mouse that had this cool phenotype, and you done all this great work with it that somebody else made. Well, that's good luck. But what did you bring to it? Are you just characterizing something that somebody else started? Or did you really add value to the project? Are you a good thinker? That shows up not so much in the seminar as it does in talking to people when they ask you about your work or what you think of other work in the literature, or they tell you something about their work. Can you listen to what somebody across the table is telling you about the work in their lab, and then ask intelligent questions on the fly about, have you done this control, have you done that, what did this thing, how does this fit with Smith's findings that they just published in Molecular Cell? Can you discuss science interactively? Are you familiar with the literature? If somebody asks you a question, can you come up with intelligent answers that show good scientific thought? Okay, so before you go, you need to practice what you're going to do. And that means practicing the seminar, and not just in front of your lab. First, one thing, they've already heard you give this talk 5 times. Secondly, they already know what you do, what you think, what the boss thinks you think, what the boss agrees with that you've done, all the terms, all the methods, all the assay conditions. And the people in the audience you're trying to impress don't know any of that. So if you just practice it with your own lab, it's not going to be very helpful. Get some friends that you know and dragoon them into coming. Now, what do I mean by that? The talk you're going to give for a job needs to be understandable by a reasonably well-educated scientist, not just a specialist. So I consider myself to be a really reasonably well-educated, reasonably broadly experienced scientist. I ought to be able to understand your seminar pretty much no matter what the topic is. And because I might be on the search committee, you'd better figure out a way to bring me up to speed so I can understand it. Okay? So it ought to be understandable, which I just said. Practice your delivery. Now, there's one really good reason why it's important to practice, because sometimes when you get up in front of an audience, you can't bring yourself to finish your sentences. There is nothing more distracting than that. So figure out a way to practice it. Now, there are various programs that you can use that allow you to record your seminar as you give it on the screen. Almost everybody these days has got access to a camcorder or an iPad or something that you can do it. But practice it so you get to the point where you don't have those funny speech impediments. And the other thing that I want to point out is to always practice with the pointer, because the other thing that is really distracting is when somebody turns the pointer on and then says this and then this, and here's this lane and here's this lane, and this gel did so-and-so, and then these cells, note the nucleus here and the cytoplasm over here. Point to what you want to have them look at and then stop. Then point to the next point. This band was more intense than that band, from which we concluded that the following was the case. These cells were stained with this antibody, and these cells were stained with the control antibody. Now, if you are nervous, that's why you have 2 hands. If you have trouble controlling the pointer, put it between you and hold it with 2 hands. You want to appear calm, and you don't want to distract them with something like this lightsaber business. This is great for Luke Skywalker and Darth Vader. It's not good for you. All right, so preparing the slides. Simplify and linearize. It is time and time again, I see somebody putting 4 or 5 panels of information on the same slide, only a couple of which they're going to even refer to. You need to remember that when you throw up a slide that's got 4 or 5 panels on it at the same time, there's the time course in the upper left-hand corner, and there's an electrophysiologic, single-cell patch clamp recording on the other thing, and then there's a gel, and then there's a micrograph or something. You have no idea what the person who's in the audience is actually looking at while you're talking. Okay? You want to spoon feed this in a linear, intellectually relevant sequence. Okay? And one of the things to do is to bring everybody up to speed on that, because you're trying to educate somebody about complex work. So images and stuff that might be fine in a 8-panel figure 1 of a paper in Cell is not appropriate to give in this type of a talk, because you just wither them with information, most of which may very well be irrelevant to the point you're trying to make. So you need to delete those issues. If you put your talk together, which is what I always do, I might end up with 70 slides when I'm preparing a talk. And then I start going through it, and then I realize that this is really a side issue, where I only spent 15 seconds on this slide, out. You do not want to throw a bunch of slides in there and then click through them and not actually use them. All right, so a good idea is to, at least to bring everybody up to speed, is to be able to use procedural cartoons of some sort which define the method you're going to use, or the primary protocol that's involved in all your experiments. And there, you have to realize that the experts in your field who already know this, already know what this cartoon is going to show. But that's not the audience for the cartoon. The audience for the cartoon is everybody else in the audience who never heard of monocratic diffusion, or don't know how a microencabulator works. Now, I just made those 2 terms up, so I don't know how either of those work either. All right? So you need to skip the tiny details unless they're critical, or if you're the one who developed them. So if you figured out some cool new wrinkle, then that might be something you want to make sure is there on the slide. Here's an example of a slide. This is from a colleague of mine named Mitchell Azar at Penn, and it's a number of years old, but it makes the point. He put together this was a review article in Nature, and it talked about members of the nuclear hormone receptor family, SXR and CAR, which are liver xenosensors that detect the presence of environmental substances and turn on all the drug metabolism enzymes by increasing transcription and translation of all 3 types of drug metabolizing enzymes in the liver. All right, resulting in conversion of the chemicals to metabolites. Well, I don't know how many of you have ever heard of SXR or CAR, or anybody who works in here on drug metabolism, probably very few of you. But in just one little slide, I brought you up to speed to what the heck this field is all about. So now in your own brain, you can say, gee, I wonder what happens if you use siRNA to knock down SXR levels. Or what happens if you knock down levels of UGT1? Or what happens if you overexpress this P450? Well, which P450? So just by giving you that, I've set the stage for a whole variety of things that I might want to talk about, and you're already thinking, oh, I understand, so now I see what he did. All right. So, next thing you need to do is to review what the data are you're going to present. And there are a couple of points I want to make here. First of all, you don't want to try to convince them of something which is patently untrue based upon what you're showing them. A great example of that is to show them a bar graph with some standard error bars that overlap. And then you say, oh, the slight effect that's on something. If you say that and the confidence and the 95% confidence limits overlap, standard error bars overlap, It means there is no statistically relevant difference between those two. And if you say, well, there's a slight effect, all you're proving is that you don't know any statistics. That is really dangerous in an academic setting, and it's a death sentence if you're looking for a job in a pharmaceutical company or a biotech. Okay? Gel bands that are not different to the eye can't be explained away by saying, well, it doesn't really show very well in this particular gel. But this is 5-fold more intense than that. Come on, I already know you picked the best gel you've got. Don't give me that crap. So if it's not evident to me, don't try to convince me of what you're going to say about it when what I'm looking at with my eyes is simply not consistent with that. There are two phenotypes of people in the audience. Those who believe you when you're trying to do that sort of stuff, or when it's not clear where the conclusion comes from in the data, and those who think you're lying. I happen to be in the latter group. If I can't understand what you're saying, or I can't read the labels on the figure, or anything like that, I just assume you're lying. Now, you might get an audience that's all people who say they're all full of sweetness and light, but there might be an SOB like me out there who's going to say, wait a second, they were terrible. Okay. Companies want to see dose response curves and error bars. It's not just a one-off experiment in which they clearly haven't repeated it. The next thing I want to tell you is to review the human disease context of your work. Particularly if you are looking for jobs that are going to be supported by NIH grants, which almost everybody in the health sciences does these days. The NIH doesn't fund studies of yeast liver. The NIH is interested in humans who get sick from something and how we understand it. Now you can use yeast to look at it, but you'd better have some understanding of what this gene's ortholog, what this yeast gene ortholog does in the human. Who gets sick when this gene has a mutation? Okay? So here's an example of some dose response curves from the work that was done by my group years ago. But the point is, it's got dose response curves, it's got error bars around the points, and it's clearly labeled so you can see exactly what's going on. So that's an example of a slide that allows you to talk about the efficacy of whatever this experiment was. Whoops. In this case, this goes to a higher level than that does, so the efficacy is different, and the potency is different. The half maximal concentration is different. All right, here's an example of bar graph. This was an experiment that was actually done in Ron Evans' lab at the Salk, so that's not my work. But the point is, you can take a look at this and you can clearly understand whether there is a gender difference in the experiment, and what happens if you use various compounds in the experiment. So try to simplify the experiment. So you may be going on to much more complicated stuff, but it's better to pull it across one piece at a time. Now, the next thing you need to do is you need to check for antagonism. Now, what do I mean by that? Well, there's going to be some hotshot there in the audience who's trying to try to trip you up, because if they can trip you up, they're going to be able to prove to their boss that they are smarter than you are. Furthermore, you may be going someplace where another bunch of people work on the same thing that your boss's lab works on and that you work on, and they may disagree with the whole concept of what you guys think is happening. Well, you better know that going in, so you know what you're liable to be asked when you get there. All right? So that's very important. Now, sometimes they may, everybody, maybe sweetness and light, everybody agrees this is how life works. But you better think about that before you go. Next point is you need to make sure that you and your boss have had the discussion about what exactly you're going to give away. If there is some sequence or some something or other that you guys have found, you haven't published yet, or some cells that are the key to being able to work on this, or some mouse line that you got from so-and-so, you better make sure that both of you are in agreement on whether you're going to divulge this information or not. And maybe yes, maybe no, but don't come back and say, oh, I told them about so-and-so, they were really excited about it. And the person, your boss says, you what? So, this has to do with whether you think they're going to be in agreement with you. Now, your bunch, you may be doing something which is so cutting edge that the reason that that place is interested in hiring you is specifically because you have really pushed the envelope. That's the best position for you to be in when you're looking for a job, is if they think you are really a mover and a shaker in this field. But you better be prepared that they're going to be skeptical about this. And they're going to want to see how well you can defend your positions on that. Do a quick check for late-breaking articles before you do that. And the reason for that is that all the people that are in the audience will have been doing that. And you don't want to find out that a paper just came out yesterday or last week in Nature on this thing and you know nothing, you don't know anything about it. Well, not everybody can be up to speed on every article, on every topic. But at least, you know, Nature, Science, you know, whatever your top couple journals are for your work, try to make sure that you have some idea of what's recently been going on. And the reason is they may say, how does your work compare to that of Berkowitz and colleagues reported last week in Molecular Cell? And you're sitting there saying, duh. Now I want to talk about putting the talk together and how to deliver it. You start out, in my suggestion is for about the first 10 minutes or so, you need to say, build the scene. This is where the field was before I got into it. I came to my boss's lab, and at that point, they had shown that this was the story. And so she suggested that I look at this aspect of that problem. And so we developed this project around trying to test whether what they had already found was true under these circumstances. And so on. Well, you weren't there then, so they did this, they did that. The field was so and so. All right. So that's how everybody fits into that. Okay, so now I've arrived. Now you talk about. So I took this project, and here's where it's important that you switch to the first person singular. I did the following experiment, and when I did it, this is what I did, and this is what I concluded from it. And on the basis of that conclusion, I reasoned that if I then did this experiment, it ought to come out like so-and-so. Next slide. Here we see that indeed, as I had presumed, it did exactly what I had predicted. So on the basis of this, okay, another postdoc in the lab had developed a cell in which they had knocked in some SNP allele for this particular protein. So I got her cells, and I repeated the experiment. And now, I reasoned that if this gene was downregulated or had this allele in it, the cells would respond differently. So I did that experiment, and here are the results. Click. All right, this is how I thought, what I imagined. But it also builds in, I collaborated with so-and-so who had this reagent. We did this together, or she had already done this microarray, and so we picked the 5 top genes that were upregulated and the 5 top genes that were downregulated, and we asked whether or not these were merely markers of the process, or whether or not these proteins actually had a role in controlling the phenotype. So that's what you decided to work on, what you found, how you interpreted it, where you went next. And then you say, so this is where we are now. Now we know that this pathway, whatever, this is where it's a good idea to have some conceptualized model that you can show how this pathway functions. Because now you're going to go on, and in the last few minutes you're going to say, so what's really important to do next? And this is what I've been planning to work on, that I've written a K-99 grant on, or got a K-99 grant on, or whatever. What really needs to be done here is the following. Now we need to, I've done it in these cells, but what I haven't done is put them back in the context of the animal. So now we have to knock in and make a transgenic mouse that has this particular allele knocked into the authentic locus. If we do that, then we predict that the animal will have the following renal dysfunction, okay, which we're going to look at by whatever. Now, most job interviews these days involve two steps. The job seminar, which is what I'm saying is generally given to everybody, and then a chalk talk, which is given just to the search committee. And particularly if it's an academic job where they want to know what you're going to work on, they're going to want to know what you're going to do. Well, if you've already gotten a K-99, or even if you wrote a K-99 grant but didn't get it funded, you've still got the document that says this is what I plan to work on, and why it's important, and how I'm going to do it. And almost every time, the search committee's first question out of the box when you walk into that chalk talk is this. They look at you and you've got your little piece of chalk or your magic marker up there, your marker, and they say, okay, suppose we hire you and you come into the lab. You come into your new lab, you show up on the first day. What's the first experiment you're going to do and why? Never thought about that before. Well, I'm telling you to think about it, because what you walk in, here's the technician, here's the postdoc, and he says, what am I supposed to do? Well, what are you supposed to do? All right, so during the seminar, make sure that it finishes on time, okay, because everybody, if you go to a noon seminar at 1 o'clock, everybody, you included, are fidgeting to get the hell out of there, okay? Companies often will squeeze you to get every ounce of information out that they can, particularly if you come out of a big lab. It's a major mover and shaker in the area of atherosclerosis or something. They will stand up there and they'll have you up there asking questions until they, what cell lines to use, what conditions, how long does it take for this to show up? If you stress the animals with phenobarbital first, what happens? Does it get worse or better? How do you measure it? What instrument do you use to measure that, etc.? People usually will interrupt to ask only pointed questions. Now, what do I mean by that? Generally, the host will ask you in advance whether you'll entertain questions during the talk, or very often there will be a tradition that they'll say, let's let so-and-so give their talk, and then there'll be time for questions. So generally that contract is decided in advance. So if someone does ask a question, you have to decide on the fly whether that's something you're about to come to or whatever. Don't give answers unless you are sure. What do I mean by that? Well, suppose I ask you some question out of the audience about your work, and you guess, and I say, what happens when you do so-and-so? And you say, have you ever done that? And you say, yes, we have. Then chances are I'm going to say, well, what were the results? And if you didn't actually do the experiment, I promise you that is not a friendly deal to be in. Another point is, don't argue. If somebody raises a good point about a lesion in your data, you haven't looked at, have you ever tried this in, cells that were null for this, or have you ever done this in cells that have been starved, or that were, are these randomly proliferating cells? Yes. Have you ever done it with synchronized cells to see what stage of the cells have to be in to show this response? Well, you know, there are two ways to go. One is to argue with them, and that would be a dumb experiment. Okay? The other way is to say, that's a good idea. No, I haven't. I had to try that. All right. If they ask if you've done something, give the answer. Don't try to snow them, because the next question, as I just said, is, so what were the results? Last point here. You can't know everything. So if you don't know something, don't try to fake it. It's fine to say, I really don't know. It's better that than trying to weasel your way out. As an audience can, I mean, I'm sure you've had experience where you're sitting listening to somebody answer it, and you come to realize this person is clueless. They're just trying to BS you. All right, so getting an academic job. First of all, the search committee has been formed by the chairperson to try to bring somebody in who has some hotshot in. So what are they going to ask? First of all, they're looking for somebody who's going to be competitive in science in that field. Are you going to be a hotshot? They don't want to hire some jerk. Okay? Are you going to bring a technology or be able to anchor a core facility that will bring new methodology into the department and a new core facility that they can use? So it's really good if you have gone around and done a little homework on what everybody does and say, well, you know, I use this so-and-so method, and I was talking with so-and-so this morning, and that's the type of cells in which we can actually sort them on the basis of this phenotype. And that doesn't matter. I mean, there are many examples. You're going to need to teach a required course, especially if you're in a medical school. So that means, again, practicing your delivery, trying to get opportunities to give as many talks in your current spot as you can is always good. Don't try to avoid them. Try to give them, because the more at ease you can get to be, the better off you are. Now, teaching, particularly if you can teach a core basic science, portions of a core basic science course in a medical school, that's really important. If you can teach gross anatomy, you've essentially tenured yourself, okay? All right, and because a lot of courses are team taught, that doesn't mean teaching your own specialty. You might give a graduate seminar in your own stuff, but first and foremost, they want to see the core stuff taught. And if you're in a medical school, here's the catch. If you try to get too sciencey, the first-year medical students are going to complain that that stuff doesn't have any clinical relevance. Now, that may be the most idiotic conclusion to reach about it, but that's what they think. And you don't want them going to the dean on Monday morning bitching about your inability to teach folic acid metabolism. The dean does not want to hear about the fact that they don't like the way you're teaching folic acid metabolism, so get it right the first place. The next and most important thing is they want to know when after they talk with you, are you going to be able to write and think and plan and self-promote well enough to become fully funded within a couple of years and stay that way? Well, they want to know, that's why the K99 R00 grant mechanism is so powerful. It's because if you get one of those, you've already proved you can get a peer-reviewed grant. Not a guess, it's a fact. Now, that doesn't mean you'll never lose grants in the future, but it's a great start. Okay? Now, what about a company job? Well, what they're trying to find. First of all, a team player. The reason is because everybody's working on a different aspect of a project. You're doing this piece, but the stuff, the compounds you're testing came down from somebody in another building. You get them on Monday, and you're going to do your assay and then report the results out. And on the basis of your results, the next bunch is going to do some other assay on it. You have to have excellent scientific capabilities, just like in academics, okay? They hope that you're smart and that you're going to be able to keep abreast and not just do the same hidebound methodology 5 years from now or not understand the new technology, okay? Reliable and on time. if the sample comes to you on Tuesday and you're supposed to have the results out by Friday morning, that doesn't mean have the results out by Monday because you decided to go to Asheville for the weekend on Friday. That means Friday morning. Reliable also means ethical, honest, and truthful. That is at least as important in companies as it is in academics. In academics, we're all sort of self-driven to be ethical, honest, and truthful. And public science is intrinsically self-correcting. If you publish something which is false, it will be found out eventually because nobody will be able to repeat it. In companies, the work is secret until, for instance, for drug discovery, drug development, it's eventually submitted to the FDA. All the data are submitted. And eventually, the errors in that will come out, and that can cost your job and cost the company millions of dollars or more. You have to have good people skills. And what do I mean by that? Particularly in a company, you're going to be a part of some team, and you're not going to like all the people on your team. There may be some people on that team that you frankly can't stand. They don't care. Your job is to be able to work on that team and get the job done. If you think in your own academics here, your boss, or you may know somebody in your department where that faculty member and another faculty member elsewhere in the department or in another department don't get along, don't see eye to eye, and they don't go to each other's seminars, they don't collaborate, even though they work on essentially the same stuff. You can do that in academics. Doesn't get you anywhere. I mean, it's stupid in academics, but it's simply not possible in the company. Okay? Able to accept criticism and make corrections. Nobody's perfect. You're going to make mistakes. You're going to find out that something was not done right. The question is, are you able to avoid doing it again? That's being able to accept and make the corrections. And then, do you have future potential as a leader or a manager? At some point, you come in and you have a couple of technicians and you're running this process or whatever it is you're supposed to do. And that's basically what you're supposed to do. They say, you are going to do this in this project area. Making hangnail drugs. Okay? Well, you got a couple people. Well, if you're really good and whatever, eventually you're going to take on more and more responsibilities. At some point, they're going to want to know whether you want to become the manager of the hangnail drug project. If you do that and you begin to lead teams and so on, the next thing they want to know is whether you want to be vice president for dermatological products. Well, you don't have to do that, but that's what they kind of like to find, is somebody who can come in at one level, grow into it. Okay, homework before you go to visit. First of all, go to the corporate website. Make sure you understand what their mission is. How do they plan to make money? Okay, now particularly in a biotech, what do they do? And how is that going to really be successful? Let me give you a good example of that. Take a DNA sequencing company. Now there's a jillion DNA sequencing companies around, and some of them have got ABI machines, and some of them have got Pacific Biosciences machines, some of them have got 454 machines, whatever. They're all sequencing the same stuff. So how is any one of them going to be any better or be able to have a competitive advantage? Well, in a biotech company that's built upon starting up with some new intellectual property around the work that's been done by some professor here in this department, in this school that's been patented and so on, what are they going to do? How cool is it? Okay. So is it unique? Do they have patents on it? Is it proprietary? Or what? Okay? Who are the scientific founders? Who actually put the package together? Okay? Who's on the scientific advisory board? These are the people who ought to be the heavy sledders. Probably you ought to know some of those names. If it's some humanized antibody biotech company that's going to make humanized antibodies, there ought to be some hotshots in molecular immunology on that board. Names you probably know if you're an immunologist, okay? Then you look at the corporate board, and the corporate board is a separate bunch of people, and these are the business people. And generally, when you read that, you won't know any of their names, and all you'll see is that these people have been involved with lots of startup companies. That information is probably pretty irrelevant to you. You may see some names of some companies you recognize, but these are always kind of embellished. But it's important to know who actually sits on these things. What are the patents that they have? The best way to look for patents is on Google Patent. If you go to Google and you go to all the tools, it says map and Gmail and so on, and it says more. You open that, you click on that, and you go down, and there's a whole bunch of other stuff, so and so. And then at the bottom of it says even more. How many have done that? Right. So one of them says patents. And if you go to that, you open it up, and then you can search for the name of the company as the licensee of patents to see what they hold, what patents they have. You can look to see all kinds of things of that sort. Now, one of the things, learning to read patents is a separate lecture, but I'll tell you this, a patent has to teach the reader to do something, and what is being claimed to be able to be taught is what's in what's called the claims. So when you open a patent, you can look and see what they're claiming can be done. So that's the most important part, to see what it is they say that they're going to do. All right, now that patent search is only good for looking for issued patents, but it's a good place to start. And the interesting thing is the Google patent page starts out with a randomly selected group of about 5 patent little pictures, little icons that show up for the craziest devices that you can imagine. That, you know, apple peelers or automated baby buggy rockers from 1923 or something. My favorite is one that's called the bumper dumper. Okay? And it is an attachment for the back of a Chevy Suburban that allows you to mount an outdoor toilet on it. Do not leave here and go to Google Patent and search for bumper dumper. Do not. All right, now, another thing that shows up on a company's website is called the SEC 10K form. This is a report that's done every year to the Securities and Exchange Commission, and it talks about who the investors in the company is, what's the company's strategy, how much money they have on hand, who owns how much outstanding stock. And it's a very good place to look to see a lot about the business side of the company, of all sorts. Okay, now you're going to interview on the interview day, and I'm assuming now that you are a PhD with postdoc experience going to look for a job. Well, first of all, in a company, certainly it's not going to be more than a day, and often maybe only half. They may have you come in at show up at noon, give a talk, 5 o'clock they'll say, thank you, okay? In biotechs, there probably will be a seminar because biotech companies are earlier companies, and they generally have a bunch of recent academics, and they just take the opportunity to have somebody come in. It's good to have them give a science talk, and it's a good way to wait. In the pharmaceutical companies, big pharma, you go look at a job at Novartis or Merck, Bristol Myers Squibb or something like that, they may not do that. Keep in mind, you may be taking somebody else's job, and they may be there in the audience and they may not know it. They may be one of the people who interviewed you. This actually happened to me. I was going to a company, first company job, and I was coming because the guy who was doing the job wasn't doing it well enough for them, and they were going to replace him. And he was one of the people that I met with. It was a little strange. Now, you'll meet people at all levels. What do I mean by that? When I was in a company, I had a guy who ran our protein overexpression core laboratory who had a master's degree in biology or some field like that. And he was the best interviewer I've ever met. He could size somebody up. He was amazingly good at intuitively figuring out whether somebody was likely to stay, whether that person had really been the one who had all the ideas. He was great. I never tried to interview anybody that Dave didn't participate in the interview. I'm a great recruiter, but I'm worse at assessing somebody's capabilities. So you're going to be interviewed by technicians, by postdoc, and by junior people, by staff or whatever, and you generally be done. And almost every time in a company, the last person you'll talk to will be somebody from human resources who will sort of tell you about the benefit package and the opportunities and things like that. It's likely that they're not going to take you out that dinner. It's not like when you go to look at a job at a university where several people take you out and get you drunk. Generally, there's not even a dinner. It may be, it's certainly not fraternity rush. All right, so they will collect opinions in writing from everybody who interviewed. And then the hiring person, they will try to take a look and rank you to see what's going on. Now, who's going to be interviewing you? Well, first of all, your direct supervisor. Second is, as I told you earlier, some hotshot from your area who the supervisor said, Freddie, I want you to go and listen to Schrader's talk and let me know what you think of that stuff. And that person makes their bones by asking you a bunch of rough questions. Other team members, as I said, some junior, and then a human resources person who generally sits through these interviews. Now in the government, for instance, we always have a human resources person there in the room for every step. Not because we want their opinion, they're there to make sure that nobody breaks the law by saying something dumb. Now what do I mean by that? We aren't allowed, not only in the company, any place, it's not allowed to ask about your marital status, your health, whether you have a significant other, whether you have children, nothing, unless you raise the issue. I'll come back to that at the end. All right, so you've been in the company. I already talked about what the search committee wants for a university job. So that's what I just said for the university. Well, what about the company? Well, in the business, first of all, they still want you to make sure that you know a competitive scientific field. They want you to anchor in technology, just like in academics. They want you to be able to communicate effectively. That means giving team talks on your research, putting together PowerPoint, going to a collaborative meeting with Wyeth or Merck or whatever, and making a presentation. I gave a talk one time at an research alliance that we had with Bristol Myers Squibb, and it took 2.5 hours. There were 120 slides, and it took me 2.5 hours to give the presentation. Okay? Get along in a team environment, as I described. And last, being able to meet timelines, and then do work that makes money for the shareholders. This is important. It doesn't mean publishing lots of papers. Companies don't care whether you publish the papers in general, except in very early startup companies. Once they get rolling, pretty much at all, money is tight, time is tight, and they need to do stuff which is related to producing whatever the drug or the products are that you're supposed to be working on. If in the time remaining you want to do some work, otherwise, that's cool, that's great. But that's not why you're there. All right, so you're going to be interviewed. Well, are you versed in the field? Can you discuss it? All this stuff is exactly the same as it is in academics, okay? Except for this one. Are you familiar with the mechanism of action of their drugs or technology? So if this is a company that makes humanized antibodies for the treatment of rheumatoid arthritis, you ought to be familiar with what that drug landscape. What kind of drugs do they make? What's out there? How do they work? Things like that. On the company side, they want to know, are you either under or overqualified? Well, what do I mean by that? Well, if you are if they're bringing you in for kind of a menial level job, they're going to say, this guy isn't going to stay. This is not going to be challenging. So they either have to decide how they're going to, if they really like you, they can try to tweak the job around in some way and say, well, we want you to start on this, but then there's this other area that's opening up. Or you may be overqualified, and this person's not going to stay. She's going to get here and she's going to be bored as hell. So as soon as she walks in the door, she's going to start shopping her CV around. Okay? Are you a team player? Notice how this keeps coming up. Okay? Finally, do you look like one of them? And what do I mean by that? I had a really bright friend of mine who was at UC San Diego, who was a postdoc there, who was a really, really smart geneticist. And he was out looking for jobs. Well, his hobby, I've known him since he was a graduate student, his hobby was racing motorcycles. And so he had really long, black, kind of greasy hair that he looked sort of like one of those guys out of some biker movie. And he wore black leather jackets with silver studs on them, and old blue jeans and stuff, and he rode the dirt bikes around. And he usually had about 2 days beard growth. Well, he would scare the bejesus out of somebody in a lot of companies. They'd say, "This, good grief, we don't have to touch this person." Now, I know that's silly, but think of, think about an understated wardrobe, okay? Now, do's and don'ts. Be sure to ask about their published work. Make sure that you are going to meet your direct supervisor and find out about what opportunities there are for advancement. If you're going to come in and do something, what are you going to do next? Don't ask about their non-public work stuff. They're not going to show you the structure of the compounds they're working on unless you've signed a confidentiality agreement. And they're going to be reluctant to do that. They won't tell you how close they are to having a drug to come to the market. Okay? They won't discuss models. They won't tell you what cell lines they use unless they've already published it. So compared to academics, you walk into somebody's lab or whatever, and they'll tell you everything. They'll open up the notebooks. Look what we did yesterday. It does not happen in a company setting. All right. They're not going to keep the job open in the company while you make the next knockout house, and because you've got a lot of work to do. It's open for a period of time, perhaps a quarter, a few months. Now, at the end of that few months, if I'm the hiring person and my boss, the VP for research, has given me this job slot to fill, and in fact, I haven't filled it in 4 months, he's going to say, well, Schrader, you've been doing your work fine, and you haven't needed that head count bad enough to fill this slot, so I've given it to Freddie over here, who really needs it now. So that's it. You lose. Okay, don't pick over things like the size of the office or something like that. That's fine in academics, but that game doesn't wash in companies. All right, what they particularly don't care about is your willingness to stay forever. If you are a tenure-track assistant professor and you get a job at Duke in the chemistry department or cancer biology, whatever it is, if you do well and get grants and whatever, you probably are going to be happy to try to stay here until recruited away to something really cool. Maybe never leave. In companies, the average time to stay at a company is 4 years. So they're not expecting you to stay forever. They hope that you do, but people move all the time. All right? Your outside life. They're not going to deal with too much with flex time. If you get really aggressive about that, that doesn't work. There's always ways to work out flex time, but don't make it a bargaining point. Okay? They'll always deal with, you want to come in at 6 a.m. to avoid the traffic, no problem. But try to get a sense of things, first thing is to convince you you're a good scientist and get the job. You can't have any other business dealings. Okay? They don't care about your other scientific interests. If you want to work on something on the side, that's fine, generally. You generally have enough resources to do that, but they're not, you have to clear everything to be published, and you better jolly well have everything you're supposed to be done. You can't bring stuff with you, reagents, clones, protocols, nothing. That all has to be done legally by a materials transfer agreement. So they'll send you an offer. It'll come by FedEx in the next couple of days. They'll give you an exact description of everything that they're proposing for you, and they generally say this job is open for a period of 2 weeks, or whatever it happens to be. Okay? Now, you can extend a little bit. You can say, well, I've got another job interview that's for 3 weeks, and so could I get back to you by March 1st or whatever? You know, generally, they'll say, fine. You can clarify uncertainties. Are they going to pay to move everything that you've got, or are they going to give you just a lump sum and you've got to figure out how to do it? They're not going to give you a bonus with, if you're a drug company, medicinal chemists get bonuses. It might be in certain circumstances you do something that's so cool that they really need you. But if you're your standard cell and molecular biologist or animal physiologist or pharmacologist, you don't have to worry about that because that's not going to happen. And finally, this is my last slide. Don't screw up. All right, with that, thank you, and I'll take questions. I bet I know where to push this. Wow. Okay, any questions? Yes?
You said in the beginning not to get too hung up on your own field of jargon and all that. So how about on your resume, just to get through the door? Do you want to hype up your own scientific expertise with all of the equipment that you can use, or?
That's a good question. So it depends upon whether it's, if companies get deluged with one-page resumes from people that list all of the techniques they know how to do and all the equipment. It says Microsoft Word, Microsoft Excel, Microsoft PowerPoint, gel electrophoresis, and just withering, comma, and everybody knows that. It's like saying I know nouns, verbs, adjectives, and adverbs, and I can put them together into sentences. So I don't think that's particularly important to add that sort of stuff. I think that, first of all, unsolicited things to write to a company and send your resume is completely worthless. It goes directly into the circular file. If you know somebody there and you can get your boss to send that person your CV, your full CV, and say, hey, Harriet, I've got this really hotshot postdoc who's finishing up, who's done all this really cool stuff we've been publishing in FATSEB Journal. She's looking for a job now, particularly in the Los Angeles area. Have you what you got? That's great. As far as the jargon part is concerned, the jargon words show up in the titles of the articles in your papers. On the list of all your publications. It's going, someplace in those titles you've used that acronym. So, and if it's somebody who's reading it who actually is familiar with it, that's fine. I was referring to the problem of using jargon in a talk where the people who are not in your field haven't a clue what that phrase means. Anything else? I can hear you breathing out there. I know you're there. Okay, well, good luck.
Can we